Hey guys, it's Tiffany Martin in Medical Microbiology, and we are working our way through Chapter 16. We had just finished T cells, and we want to talk about MHC. And we talked about how these T cell receptors were formed, and they had these variable regions, and this is where the antigen binding site fit into that. And they were very distinct, one pair of pants, one pair of shirt, a shirt, a pair of shoes. They were put together so that they can be unique, and every T cell has a unique one that's unique from every other T cell. All right, but it can't just find an antigen just out in the body. It has to have that antigen bound to something, and that's where the MHC complex comes in. This is a these are major histocompatibility complex, MHC, and they are glycoproteins that are found in the cell membranes of animal cells, most vertebrate animate cells, and their job is to hold and position that antigen, that antigenic determinant, that piece, for presentation to the T cells. So what that looks like on a picture is going to be your body cell, just any cell here, your cytoplasmic membrane, and then you have this protein that extends out onto the outside, and it also has an antigen binding site. Now this one is just a general one. It'll hold lots of different antigens. It's not looking for a soulmate or a specific antigen. Okay, so let me go back to my board and show you that. Hang on just a second. Okay, so I'm getting my whiteboard, and I have my T cell that I drew on here, and that T cell has a T cell receptor from there, and we know it has an antigen binding site, an area where that antigen is going to bind. Now this T cell receptor and its antigen binding site is designed specifically for this one antigen. It only will bind this one antigen. Now there's other T cells. They have slightly different shaped antigen binding sites, and they will bind to other T cells. But this one is looking for his soulmate. Now he can't find it out on its own. It's got to be presented by an MHC protein. So we see this MHC, it's holding the antigens, and it holds lots of different antigens, not just a specific one. But this time he has found this one, and so as he is holding it out, and this T cell receptor will bind to that antigen and form a complex from that. Okay, so that's what's going to happen initially. This T cell receptor is going to bind to that soulmate antigen presented by this MHC. Now, all cells in the body can have this MHC protein. So it has the ability, say this body cell got invaded by a virus, it has the ability to take pieces of that virus and put it on MHC to show the T cell receptor. So that's what's going on. Now the T cell receptor will also, and that's where this line came from, will also bind to the actual MHC. So not only does it bind the soulmate antigen, but it binds to a piece of that MHC, which is why it's important to have that antigen presented on an MHC. And when you have problems with that, you have problems with your immune system, and that doesn't work. And it doesn't work well. So going back to here, histocompatibility proteins, they're found on the cell membranes of animals. Their job is to hold the pieces, the epitopes, the antigenic determinants, to show the T cells. Now there are two types of MHC. MHC class 1 are found on all the cells in our body. Epithelial cells, neural cells, muscle cells, every cell that has a nucleus has an MHC class 1. Now red blood cells are made and they lose their nucleus, and so they actually don't have MHC class 1, which actually makes diseases of the red blood cells kind of hard to manage because they don't have an MHC1. They can't tell you that they've been invaded. So that's kind of interesting. So all cells have an MHC class 1. There are certain cells that have an MHC class 2. Cells that are considered antigen-presenting cells. You remember our dendritic cells, dendrites? Those dendrites were part of our skin and mucous membranes in our innate immune system. And those dendritic cells, their function was to identify and phagocytize foreign bacteria. So a pathogen would come in, the dendritic cells would swallow it up, phagocytosis, eliminated by exocytosis. But you remember I said it saves a couple of pieces? This is where that comes into play. It saves a couple of pieces on this MHC class 2 and then tells the rest of the immune system, the big guns, our third line of defense, hey, this is what I found. Do you think this is bad? That's what it's doing. So dendritic cells are antigen presenting cells, so are macrophages, so are B cells. All of them, they're cells, so they have class 1, but they're antigen presenting cells, so they also have class 2 proteins involved with that. So let me tell you a little bit about how those antigens and those epitopes get put on an MHC protein. So this is called antigen processing. So here is my dendritic cell, or a phagocytic cell. It's an antigen presenting cell. It's capable of phagocytosis. So it has class 2 MHC. It also has class 1 because it's a regular cell, but we're going to look at how it presents it on its MHC class 2. So it phagocytizes a foreign pathogen, comes in, and you know that has the vesicles, and those phagosomes and those lysosomes will fuse. Well, there's also MHC2 proteins in vesicles, and they will also fuse with those phagolysosome. Remember, phagosome, lysosome fuse, you get killing reactive chemicals. These will also fuse, and now I have all of these little bits and pieces that are being chopped up and killed in relationship with my MHC proteins. And so some of those will get bound to these MHC proteins. Now we know the last step of phagocytosis is exocytosis, elimination. So they're coming out here. It will fuse with the outer membrane, and now I have on the outer membrane of my phagocytic cells my antigens presented on an MHC class 2. Same thing happens with a class 1, or a very similar thing. So here is a class 1 cell. This is just a regular body cell, looks like an epithelial cell, minding its own business, and it gets invaded by a pathogen. Something comes in, and now I have a pathogen. Well, the endoplasmic reticulum is also capable of making these MHC class 1 proteins. Remember, immunology is not its job, so it just has class 1. Pieces of those pathogens, the antigens, the epitopes, get chopped up and placed on an MHC class 1. It moves through, makes a vesicle off the endoplasmic reticulum, moves and fuses with the cytoplasmic membrane, and now here's my cell, my epithelial cell that has been invaded, showing the immune system, hey, this is what's inside me. Can you do something about this? So that is how we process endogenous antigens. Remember, endogenous antigens are once it's already invaded a cell for that. So that is MHC and antigen processing. Let me know what your questions are and bring that to Zoom. Hey, one thing I forgot to say, the reason we learned about these MHC class 2 proteins, or any MHC proteins, how did we learn this happened? We learned it when we started dealing with organ transplantation. So in the early parts of this where we're like, okay, someone had skin burnt off, can we take someone else's skin and protect them from that? Sometimes it worked and sometimes it didn't. And they found that if you were closely related, it had a better chance of working. When it didn't work, your body made this immune response and killed all the tissue. So they found that the more closely related you were, the more chance it happened. And that's because your MHC is closely related. So if you have a family member, you have a good chance of having your MHC be identical. So when this complex happens, your body says, oh, that's regular, that's our cells. Here's your kidney transplant, and this must be our cells. So it accepts it better. So anyway, so that's how they figured out about MHC. When they talk about doing organ transplants and testing to see if you're a good match, that's what they're testing for. They want to know if you have a similar MHC. Their best chance is, again, family members in your same ethnic group. So there is, if you can't find it within family members, there is an organization that will maintain a list of people and their MHC so that potentially you can match up. So I'll put a link to this and check it out. It's kind of interesting, but that's how MHC works. So bring me your questions and I will see you at the next video.